Alright, so this video is going to cover cell transport. And so we've learned about the cell membrane. The cell membrane is a selectively permeable barrier. And so what that means is it kind of regulates what goes in and out of the cell. And there's two main types of transport that are going to occur, and that is either passive or active. And if you just kind of think about what those words mean, it should be kind of helpful for you. If you're being passive, it means you're just kind of chilling. You're not really doing a whole lot. But if you're being active, that means you're up and moving around, and in order to be active, you need energy. So passive transport is a type of transport that does not require energy, whereas active transport does. And the reason why is it has to do with the movement of the molecules. So if you think about uphill and downhill, it really doesn't take a lot of energy to go downhill, but you do have to put in a lot of effort to go uphill. And that's essentially what we're talking about with our molecules. So in a cell, remember that you have a solution, and then that cell is also surrounded by a solution. And so we look at the concentration of molecules inside versus outside the cell. And if there's a difference, we call that a gradient. And so things will naturally like to move from where they are higher to where they are lower. And that's what we call passive transport. Anytime a molecule is moving from an area of higher concentration to lower concentration, it's going to be passive, and it doesn't require energy to do that. We call that moving down the concentration gradient. So the key to passive is no energy, because we're moving high to low. We're kind of have that downhill movement with the gradient or down the gradient. Now, active transport, on the other hand, is going to require energy, usually in the form of ATP. And the reason why is because we're moving molecules from where they are lower to where they are higher in concentration, and they don't naturally want to move that way. So it's kind of like going uphill or up the concentration gradient. And so we're going to look at some different types of both of these. So in passive, there's three main types. We have simple diffusion, facilitated diffusion, and osmosis. And in simple diffusion, it's just the movement of molecules from high to low. But in this case, we're moving directly through the lipid bilayer. Now, diffusion is just a general term for anything moving from high to low or spreading out. But in terms of the cell, simple diffusion is going to go straight across that phospholipid bilayer. And this usually is going to be small molecules that are not ions, and they're normally hydrophobic, because in order to get through those fatty acid tails, it's going to have to be a hydrophobic molecule typically, or it's going to have to be very small. So things like gases like oxygen and carbon dioxide, and some of your lipid-based steroid hormones can move across this way. And so simple diffusion is just simply moving from an area of high concentration to an area of lower concentration. So as you can kind of see in the picture, initially there's a lot of the molecules up here, and as time goes on, they're moving to where there are fewer of them, and they're just kind of spreading out. And they're going to continue to do that until they're relatively equal, and then they're going to move kind of back and forth. And so simple diffusion is just a movement from high to low. Now, the next type of passive transport is facilitated diffusion. And facilitate means, facilitate means to kind of help. And so essentially it's diffusion still. We have high to low, but it's going to require the help of a protein. So the key to facilitated diffusion is that the molecules are moving high to low through a protein in the membrane. And we typically either call these proteins channels, where they pass on through, kind of like a hole in the membrane, or they're carrier proteins, which are going to have to kind of attach and then open up and then pass through. And so in the case of facilitated diffusion, we still have a high to low movement, higher concentration to lower concentration. But this time, instead of moving directly through the bilayer like it did in simple diffusion, it's moving through one of those chunky proteins, okay? And so some molecules that do this are molecules like glucose. They're a little bit bigger. Amino acids, those building blocks of proteins, they're the type that are going to move through the big chunky proteins in the membrane. So it's still passive because we're moving high to low and energy is not required here. And then the third type of passive transport is going to be osmosis. And osmosis is specifically the diffusion of water across that membrane. So anytime there's a difference in water, which is going to be our solvent, anytime there's a difference in water inside and outside the cell, the water is going to move. Once again, it's passive, so it's going to move from where it's higher in concentration to where it's lower in concentration. And so water is always going to move to wherever there is more solute and less water. And so in order to kind of really understand osmosis, we need to understand a little bit about tonicity, which has to do with the concentration of the solute. So just as a quick reminder, in a solution, the solute is what's getting dissolved, and the solvent is what's doing the dissolving. And so in the case of the cells, inside and outside the cell, the solvent is going to be water. And so if we have a difference in concentration inside and outside the cell, the water is always going to move wherever there is more solute and less water, because water is going to move from high to low. And so in order to kind of describe solutions in terms of osmosis, we use these three terms here: iso, hypo, and hypertonic. Now, one thing you need to understand is that these terms are always being used when we have two solutions. And in the case of a cell, our two solutions are going to be the inside environment and the outside environment. And so if we were to describe solutions as being isotonic, iso means equal, so that would be the cell and the surroundings both have an equal concentration of solute. So in that case, this is the picture here. And so what you can see is that there's an arrow going out, there's an arrow going in. If a cell is in isotonic conditions, the water just kind of moves in and out equally. Now, these other two terms go together, okay? When we have the cell and the surroundings, if one of them is hypotonic, the other one is hypertonic. And hypo means lower or less solute, and hyper means more or high solute. And so if the cell is hypotonic, that means the surroundings are hypertonic. And anytime there's a difference in solute, that water is going to move from wherever there is more water to wherever there is less water. And so just kind of a quick, example. Let's kind of make sure we know what we're doing here. So in this case, we have cells. Okay, this is what the little kind of bag looking structure is. Okay, notice that the cell has a concentration of 10% salt. And so in beaker A, the surroundings also have 10% salt. And so in beaker A, we would call that isotonic because they are equal. All right. So in this case, on this beaker here, the water would just move in and out equally. Now beaker B, the cell is 10% and the surroundings are 2%. So beaker B has less solute, so we would refer to that solution as hypo. And then over here, beaker C, we have a 10% salt solution in the cell. The outside is 30%, which has more solute, and so we would use the term hypertonic to describe the solution in beaker C. All right, so now let's talk about what's really going to happen here, okay? So because water can freely move across the membrane by osmosis, it's going to be really important that the water concentrations are the same. Because what's going to happen is the more solute there is, the more water molecules are going to bind to those solutes, and they're not going to be able to move as well, okay? And so because of that, water is always going to move from where the water is higher to where the water is lower. And so water is always moving toward the area where there is more solute. Now something just to kind of help you remember this is just the phrase solute sucks, okay? Water is always going to move to wherever there is a higher concentration of solute. The solute is going to suck the water towards it because wherever there's more solute, there is less water. And so something else you can remember is hypersucks. Water is always going to move from hypotonic areas to hypertonic areas. And so in cells, this is really important because this is one of those homeostatic mechanisms, and that we want to make sure that we have water balance in our bodies, because if not, and that water goes into the cells too much or comes out of the cells too much, then we're going to start to have some bad things happening to ourselves, and we do not want that. So homeostasis is going to be key. And so what we want in our bodies is we want our cells to be in isotonic conditions. And that way, there's not a directional movement. That's what this means here. No net movement means it's not going in one direction. They will be moving in and out equally. And so in the case of a cell in an isotonic solution, the cell is pretty much staying the same, and that's what homeostasis is all about. Now, if we were to take a cell and we were to put it in a hypotonic environment, remember that hypo means less solute, okay, more water. A cell in a hypotonic solution is going to take water in. The water is going to go into the cell. And what's going to happen is the cell is going to swell, and it could swell to the point that it actually bursts open or lyses. Now, on the opposite, if we take a cell and we put it in a hypertonic solution, hyper means more solute. Remember that solute sucks. What's going to happen is the water is going to be sucked out, and it's going to leave the cell, and in that case, the cell is going to crenate or shrivel. So if you look at the pictures over here, isotonic is when those concentrations are equal. Water's going in and out equally, and our cells are going to be normal. If you place a cell in a hypotonic solution, which has less solute, the water is going to go into the cell, causing it to swell, possibly even burst. And then if we place a cell in a hypertonic solution, the water is going to go out of the cell to where there is more solute, and it's going to shrivel up. And so this is why water balance is essential. Dehydration is very dangerous. There's also such thing as overhydration. You can drink too much water too quickly. You have to drink quite a bit in a short period of time. But if our blood is not isotonic, or if our body fluids are not isotonic, then our cells are either going to be swelling and bursting, or they're going to be shriveling. And so this is why, like in the hospital, if you get a saline solution or you get an IV bag, what they're giving you is 0.9% saline, because that's what our plasma is. And so that's what we want to put into our blood vessels, because if not, our cells would be shriveling or they would be swelling, and neither one of those are going to be good things. So water balance is crucial. All right, so that wraps up our passive transport, okay? And so the second type is active transport. And remember, the key to active is that it does require energy. And so the reason we're needing energy is because we're going against the concentration gradient, or we're moving from low to high. And active transport often involves proteins, but in this case, we typically call them pumps. And that should help you, because if you have to use a pump, you're going to have to put energy into it to get the pump to work. And so some examples are the sodium-potassium pump. This is going to be really important, like in nerve transmission and maintaining resting potential in our neurons. We'll talk about that when we get to the nervous system. There's also something called proton pumps. That's an important part of like making energy for our cells. And then there's also going to be bulk transport. And this doesn't involve the proteins directly. This is actually the cell using the membrane to either engulf or expel particles. And so we're going to take just a couple of examples, like the sodium-potassium pump. Once again, this is really important in several physiological functions, like in our neurons. Now, notice here that it is called a pump, and so automatically when we see that word, we know that energy is going to be needed. And in the case of the sodium-potassium pump, proteins are always specific, and so in this case, for every 3 sodium ions that are pumped out, 2 potassium ions are pumped in. And so that's going to require energy from ATP in order to do that, because we're going to move them from where they're lower in concentration to where they're higher in concentration, and that's going against the gradient. So there is a video for you to watch over this and to take a little quiz on as you're going through the video, so make sure you do that. And then another example of active transport is bulk transport. Now this isn't involving protein pumps, but it does require energy because the membrane is having to kind of surround and engulf particles, or it's fusing with vesicles and releasing particles. And so in this case, we're moving really, really large particles, like big macromolecules, and they're never going to be able to go across the membrane or through a protein. So we're going to have to take them in or get rid of them in a different way. And it's going to involve these little membrane sacs called vesicles. And so there's pretty much 2 main types of bulk transport. You have endocytosis, which is bringing molecules into the cell, or you have exocytosis, which is taking molecules out of the cell. Now, in the case of endocytosis, it kind of depends on what you're bringing in. If it's bringing in food, we call it phagocytosis, like in the case of like a white blood cell engulfing a bacteria or something like that. If it's bringing in mostly liquid particles, we call it penocytosis. But in both cases, you have the membrane, which is kind of surrounding and creating a vesicle, and then it's going to pinch off and come into the cell. And exocytosis, that vesicle is going to be moved to the edge of the membrane, and then it's going to fuse and be expelled. And so these are going to require some energy to do that. And so there's just a couple examples of what that might look like in your cells. So this outer boundary here is the plasma membrane. Notice the membrane itself is surrounding to create that vesicle. And over here in exocytosis, we have a fusion with the membrane to expel those particles. There's a little video you can watch if you want. This is a little short video of a phagocyte, which a phagocyte is a type of white blood cell in our immune system. And what they do is they kind of ingest invaders like bacteria. And so there's a little video for you to watch if you want, or here's kind of a picture of what it might look like. There's also, single-celled organisms like amoebas. This is how they eat. This is how they expel things from their cells. But essentially, bulk transport is just moving big molecules in or moving big molecules out. All right, and that wraps up Objective 2.